Welcome to Study Time, a televised home learning program produced by Rwanda Education Board.
Hello viewers, welcome back again to the TV learning series. Today we are focusing on senior fives, but it also means that the senior sixes have a chance to dive in and be able to steal a part of the lesson. In the previous section, we looked at the genetic code, what a code actually means when we were talking about ATMs, when we were talking about airtimes, when we were talking about mobile money transfers. Why do they give you those digits that you have to fit in, and then stars and hash and all that, and then you enter? When do you access the airtime? Does it come as money? Does it come as minutes? Or it comes back as the codes they gave you, 5, 6, something like that, then space. Another 3-digit number, maybe 4 numbers, then 3 numbers, then 4 numbers. You will recall that when we were still using airtime cards, numbers or digits for 1,000 air card were completely designed differently from the airtime cards for 500 francs as airtime, if you remember that. The same applies to those that were meant for 5,000, those who were super big and are able to load in chunks of airtime, as well as 2,000. So the question now has to be, why is each airtime given a different digit as a code? Why is the arrangement of the numbers in a 500 air card arranged in different sets of numbers from the ones we would have for 5,000 or 2,000 and stuff like that? That's what we need to think about. So, when do we have these numbers for airtime, or maybe the digits for cash power, electronic bills for murugu? You feed in the digits and you find that these numbers are arranged in certain sets. What if the computer that generates these sheets of paper happens to make an error and then moves, instead of 5 digits, this time they are bringing 4? You find that the arrangement is in certain, like 4 digits, 5 digits, 6, 2, 3, like that. Now, is it the same thing that defines our bank accounts, the way the digits are arranged and then the slash and then another hyphen, then the numbers after? What if you shifted a number away from the first 0004 something and then you moved it to the other end, or you swapped the numbers? Would you access? Those who have ATM cards, what about the way you have these digits written on and you're going to make an online transfer or you're going to make an online order? How do you put in these digits? What if you make an error? Do these digits exactly fit in a certain slot they have been meant for? So today we are focusing on how a frame of the gene, of the code, can shift. If the space was meant for only +250 as a code for Rwanda, and you add there another 0, does it fit in that space? Or it moves to the next box? Why does it move to the next box? That's another interesting part. How did these people, our great scientists, find out that actually the information we have as store to be able to give us the proteins is arranged in a certain format that we look at the digits in all these examples I've talked about as an exact representation of how our digital information is arranged in us? Small clusters of data, but for a certain protein that move out a letter. What happens? 2, in these experiments they carried out, were they able to find out the truth about how the proteins we have in our body are designed, arranged, and by composition as well? The senior fives, we just talked about biological molecules, classification of proteins we have in the body as primary, tertiary, and quaternary proteins. What did you get from there that you can link up and connect to this? And then at the end of the day, we should be able to tell this coded message, what defines it? What are the true characteristics that across all organisms, they do have the same features? that's our focus today, which takes us to the background image we have here, and it is showing us some bit of data. So many letters arranged in a certain way, which we shall at the same time be able to focus on the whiteboard here. So the S6s, this should be your exercise now. Can you decode? Can you work out the formulas? Can you try to figure out how these numbers in ATMs and airtime cards are actually generated? And those who do ICT, how do you do coding? When we talk of serial generation of numbers that we have to use to install equipment, that every time you run a setup, there is always a serial number that comes up, auto-generated. Now here, we need to do this. S6s, you should be done by this time. The S5s, let's wait a little bit. If you have a brother at home, then, or sister, should be able to help you work it out. We shall come to that after. So you will check in this. We have letters, 3-letter word against a 3-letter statement in terms of capital letters. So some are the bases, as you can see. We have seen this before. But also we have like pro, we have SAR. These are short names, as you remember, those who have been studying about proteins in senior 4 and S5 as well, that's the short way to write an amino acid. So, off to what is our focus today. What were these experiments about? What were they looking for? Were they making a dictionary? English writers. How was the first dictionary designed? Kinyarwanda dictionary. Ese babyandika gute? Ijambo rimwe rihinnye rigufi rikagira ibisobanuro byinshi utanarangiza. So, now the question is, the experiments that they were working on, Francis Crick and his co-workers, they are the very people that have actually gone a mile ahead. Like we've always talked about this. We need to give them credit for what they did, because it's what we are now depending on. It's what we are using to have an understanding of our information that we pass on to the next generation. But how did they confirm that actually the human dictionary, the genetic code, is designed this way? So, 1, add a number, add a letter in certain forms of viruses that do attack bacteria. You remember these words. Can you recall the bacteriophage? A form of virus that is able to reproduce inside a bacteria, right? So funny and at the same time interesting. You want to produce, go attack another organism, enter it, produce from there, and then bust it and walk out when you're in multiples. Bacteriophages. A form of virus that is able to carry out its reproduction inside the bacterial cell. But what did they find out? And the question is, why didn't they use us? Why did they have to use some of those organisms? So one, that when 1 or 2 bases were added or deleted, the virus lost the ability to infect. You remember, like dog, push in the DNA, work out. So the ability for a virus to come and attach itself on a bacteria, insert its DNA inside the cell of a bacterium cell, and then use it to carry out reproduction. Now, what they found out was that remove one of the bases from the genetic code of this virus, the ability for it to enter a virus and insert its information there, be able to reproduce from there, got lost. They tried out another one. Instead of deleting 1 or 2, what about if we add 3 bases or remove them? So it's like, remember when we were talking about the genetic code, the arrangement of letters like we do have up here. The virus could infect the bacteria. Could. The virus could. Now, what are we talking about here? What did we see in these results that we can now be able to based on to have a proper understanding of what actually goes on when we try to distort the information. Now, that adding or deleting one or two bases caused the frame shift. Shift as in ukwimuka nyine, uva mu nzu ukajya mu yindi, cyangwa ukava mu cyumba ukajya mu kindi, ukava mu cyumba ukajya muri salo. We are moving, we are shifting. So moving the information, frame shift. So you have a frame, like we do have the letters down here. If you can look at them, let's focus on them very well. And you have this as a whole frame. And we are saying, what if we add in other numbers? Which inactivates the gene, but adding one or removing the three bases only partly affected the gene. Now let's check what exactly happens. We have these bases here, where you have a T, remember that's DNA, because it carries thymine. So we have TAGG, CTTGA, TCG. That it's RNA, after transcription, after the translation, after the extraction, remember the dogma of biology. DNA, replicate, move, transcribe, move across, translate, and here I have my protein, my hair, the nails, the whole body, all the tissues, proteins, structure, phenotype, the physiology. This can have a variety of things we can look at as examples. So let's see how they switch. They would have been T, now becomes a U. So automatically I would know that this is my RNA. Now, we are going to add or delete. How do we have these shifting? We have these letters, TAG, three letters, three letters, three letters, three letters, three bases, three bases, three bases. Now, let's get down and see. This would contain this down here. So we are now trying to have an arrangement when we talk of the triplet code, as in triplets, as in three children, or twins? No. Singles are triplets, three-letter section, as a component, as a small fraction. But for that, every other fraction we are looking at must also represent information for a certain amino acid that is going to be part of my hair, that is going to be part of my hemoglobin, that is going to be part of my muscle, all the features we display. Now, we have those triplets. Now we are going to either delete or add and then see what happens. We are adding a single base, so we are going to put G to the DNA. So we are up here. Remember, if I go back, this is where our T is. Now we're going to add G. So it's like you're writing your postal codes and you're filling an online document. So you say, home country code +250. Then you mistake it to be +2500. The question is, when you add in, does this space provided give you a chance to add in the second zero, or it will shift and go somewhere else? Remember, that's what we used as an example before. So where are we? Adding a base. So we've added a G. But what has it done? A G pushes a T, and the T pushes an A, and the A pushes a G, so we move to the next. It would also mean that even the mRNA would get displaced in a way. Now, check this out. This G we have put here is not what is here. Check that again. The G we inserted at the original DNA strand is not what we have here, but instead there is a shifting. Remember, what we see here is that the letters seem to keep agreeing. We shall only keep as triplets, whatever you do. We shall never be quadruples. We can't have 4. We shall always remain 3. So the only way to do is kick the other one out. Now, what does this mean? If it were possible that this G would insert itself on top of the T, then we would have an overlap. 2, that every triplet in DNA specifies one amino acid. We shall have to add that. 1 amino acid. So we are writing this in brief. For all of those who do take biology, you know that an abbreviation of AA is actually amino acid. So there we go. That each base is part of only one triplet. So you cannot add another layer to become 4. So 1 amino acid, that's it. 1 triplet, that's it. So what does this mean? How many amino acids do we have? Now, a non-overlapping code would require a long sequence of bases than one which overlaps, which means that there are things that just do not happen. And if they do, we still do not have any adverse effect. And that's why we're saying that replacing one base for another has little or no effect in a non-overlapping. Because you'd still code. The question now still has to be, out of the 3 letters in a triplet, how much do we need as information to be able to pick an amino acid, add it onto the chain of the proteins, and then we have our structures coming up. Now, if we are to continue with this about the frame shift, we get stuck with the very biggest question we have ever come across. What if the code was in position to overlap? Think about it. What if we had TAG, and then we also have the G on top of T, and then so you have G, then T, then A, and then the triplet is over? What would exactly happen? One, that would actually need a very short length of DNA to have a protein. Otherwise, you say, remove T, add G, you still pick the same amino acid. Like we do have as an example there. Look at it. That in T and A, TGACT, you can have these 3 provisions. Math. Multiples. How can we duplicate all those in? That's one. But what if we make a replacement with another base? So what does this mean? That out of this, we are going to replace one of them, in which we are replacing which one? This for that, and check what we would finally have. Completely different from this. That in each of the triplets we now have, everything has become different. Completely 3 new codons. And if we say that each codon is going to dictate which amino acid, then it means that, we are having a completely different protein. That's one. Because each of the codon is going to pick a specific amino acid that is different from the first one we looked at. 2, are we safe if that happens? 3, does that mean that we have new genes? And if genes dictate formation of proteins, if genes are inheritable, what do we pass on? If we completely have new structures, what does that mean? Are they preparing us for a new world, a new environment for which we have to adapt to? Is there good in it or bad? Which takes us to cracking the code. Now we need to think, how many amino acids do we have? The essential ones. 20. Common figure that is known by everyone. I think even the nursery ones are going to be told that, when you take that fish and milk and yogurt, we shall have specific 20 small structures which are going to be rearranged several times until you have your body coming up. So, if we had one base for each amino acid, then it means that out of A, T, G, and C, remember the 4 letters, would only have 4 amino acids. So the longest chain would be actually 4. What about if there are 2? Again, we go math. We would have 16, which means that we have 4 to the power 2 possible combinations. Then a 3-base code, which we now call a triplet, 1, 2, 3, will provide 4, that is A, T, G, and C, and each having a chance to make a recombination 3 times, making it to the power 4 to the power 3, in which case we can actually have an infinite number. So it means that we have more than enough combinations to consume all the 20 amino acids, which also takes us to looking at how we actually have different types of amino acids and also link them to a variety of proteins, non-ending, with varieties of functions. But the interesting part, from the same 20 amino acids, so much variance. So how do we crack this? How did these scientists come out to know that, you know what? For these 64 codons determined in each D and each amino acid, these are the combinations in which we would actually have the information. So, 1, they got a synthesized mRNA known, meaning that they were able to tell which letters there are in that mRNA, given to a medium that had got isolated ribosomes in the cell. Remember cell structures and functions. Radioactively labeled amino acids, which are going to be added, and then can be traced. That's why we label them, to know where they are and how they have been arranged. And definitely, there is no reaction that occurs without enzymes to catalyze them, to speed them up. And definitely, the ATP. So, result analysis. What about the arrangement of these radioactively labeled amino acids? How are they arranged? Can we follow the arrangement and trace it back to the mRNA? So what do we take from it? 1, that the first synthetic mRNA molecule was made from uracil as the base. So you have a triplet, UUU. And what do they get? 1 specific amino acid, phenylalanine. So from there, they say, oh, you know what? Triple U can code for phenylalanine. Noted, checked. And not until 1966, the whole dictionary was determined, which is here. That you are now should be in position to work out. We have the guidance, if we can go to the board and you focus there and be able to see exactly what's going on. What is the first letter, and then the second, and then the third? That if we are to work backwards and get up here and check it out and see if they say, first letter, we are on the extreme left, second letter, top, and then the third letter, the third, which is on the extreme right. So this is how we form the codons. That's 1, codon formation, 3-letter digit, and 3, decoding the bases to their proteins. So what do we have here? First letter, so we shall say U. Then the second letter, we pick the U, so this is 1, this is 2, and 3. So we have our here. This is our codon. So all the way through, first letter, second letter, so we would say U, C, U. And then, so you have U, A, C, and then you have U, G, and, U, G, and U. So where are we? Let's get back to the projection screen and see what's there, so that you're able to work that out all the way through. Look at the first box and check what we have. That again, if I was to go for the second, which would be a U, first, U, second, and third here, then you would have as well, you would need to check how these are arranged all the way through until we finally have it all. If we have U, C, A, and G, then this would be 1, second, and here we are. The first letter, the second, and then the third. So you have our C, which takes us to, pay attention here, that the first two are the same. They only differ by the third, in which we have a U going for a C. Did you check that? Now, the interesting part is that all these can code for phenyl alanine. Can you check that? So you complete the chart and you have your dictionary. That we finally have this. So those are our proteins. For each of the codes. So we still can generate first letter, second letter, third. First letter, second letter, like that, like that, like that, until you finish. You see, this is for the codons. The one we're having here on the projection screen is for the proteins that come out of what we just did before. Which takes us to winding this up with what exactly we see in these codes. What are the features? What defines them? What identifies them? One, universal. That we are the same. Remember the other tree of genetic life we talked about? The real ancestor of our genetic code. Two, degenerate, because more than one codon exists for most amino acids. We just gave one example. UUU and UUC all having phenylalanine, as an example I gave you. Triplet, these letters will only be arranged like postal codes or country codes plus 250. You can't add another one. 3, remember the 64 possible combinations. And 4, you have much more information than is actually needed. Some are kickstarts, like the key for the engine of the car, and others are to stop, like the press buttons to stop an engine running. And like we said, it's non-overlapping. Just seconds ago, we talked about that, so I don't have time to repeat that. And as we wrap this up with your homework, there we go. Check it out, incorporate it. I'm not going to read through. Thank you. Bye. That's your homework.